Hi, we're going to get started. I'm Shannon McQueenie. I'm the division head for Bioinformatics and Computational Biology. Normally, our seminars are introduced by Dr. Hirsch, our department chair. I believe he's luckily on vacation right now, so enjoying some time off, so I'm happy to do this. We like to introduce the students, and we ask them to write a bio, and I usually try to wing it, but I actually really like what Vincent wrote, so I'm going to read it because it's pretty awesome. So Vincent earned and this is Vincent Caruso who's speaking today. He's also defending, this is our public defense of his thesis. Vincent earned a Bachelor of Science in Electrical Engineering from the University of Southern California. After a short stint at Aerospace Corporation and a long travel break, he earned a Master of Science at Columbia, also in Electrical Engineering. This led him to decide finally not to become, after all that education, an electrical engineer. Instead, he learned carpentry. This is the part I love. This is why I didn't want to wing this. You didn't want me to wing this. And worked in residential remodeling for 7 years. A radio lab, which is one of my favorite programs, episode on genetic engineering inspired a new fascination with biology, and after 2 years of a prereqs at PCC, Vincent came to the bioinformatics program here at OHSU. His current research is on data processing and analysis for the study of human microbiomes. He's also interning, and this is really wonderful, at the Oregon State Public Health Lab, where we have a joint internship program, where he works on whole genome sequence analysis of pathogenic bacteria. So it is my pleasure to introduce him. I want to make a couple of comments. We are live streaming this, and so there may be questions that I'll be reading. We'll be asking, why am I asking so many questions? It's because I'm actually monitoring from the web. We sometimes get questions that way. And at the end, because we're recording, we'll need to use the microphone, correct? Or are we using? Yes, great. Thank you, Lynn. So just raise your hand. I'll bring the mic, so that way it's actually captured so that everyone can hear it, and people who watch the recording later can hear it. Vincent, it's all you. Thank you.
Thank you. Is that better? Oh, yeah, I can tell. Wow, that's loud. Okay. Okay, so as I was saying, microbiomes are found all over and inside the human body. And so, as you can see here, they're found in our guts, on our skin, in our mouths, vaginas, urinary tract, et cetera, et cetera. It seems like just about everywhere researchers look, we're finding microbial communities living there. So, why do we care? Well, one of the big motivations for studying microbiomes is that we found that they have a pretty big impact on our health. So a lot of studies have found that changes in what's considered the normal or healthy composition of the microbiome is associated with a wide range of diseases, and I'm showing some of these here. They range from things like Crohn's disease to acne to pancreatic cancer even. Microbial communities also are known to play a role in regulating the immune system. So microbiome community profiles have great potential to be used for diagnosis and prognosis of diseases, and they can also possibly predict response to treatments. And if you guys listen to NPR, you might have heard a story a little while ago about a study that showed that microbiome composition predicted people's response, some cancer patients' response to chemotherapy. So in contrast to the sites that I just showed you on the last slide, there are some sites that are home to a low biomass microbiome. And that just means that there's a lower density of microbes there. So some examples I'm showing you here are the urinary tract and the mucosae of the lungs. But just like with those other high biomass sites, there are also a number of diseases that have been found associated with dysbioses of the microbial communities at these sites. But because of the relative scarcity of microbes in sites like these, they present some special challenges to studying and researching them. And just sort of a hint of these challenges, both of these sites were long believed to be sterile because we couldn't grow any of the microbes there. They were so sparse, so we didn't think there was anything to study. So currently the most popular or the most common technique for studying microbial communities is to sequence what's called the 16S ribosomal RNA gene. One reason for this choice is that the 16S gene is found in all prokaryotes, so that's bacteria mainly, but it's not found in eukaryotes like humans and animals. So if you target the 16S and sequence that, then you only sequence the prokaryotic organisms, but not the human hosts, for example. So the other reason for targeting 16S is its somewhat unique evolution. And so the graph here is showing the relative conservation along the length of the 16S gene. So showing the length of different regions of the 16S gene here. And the peaks here that are highlighted by pink bars, these are the most highly conserved regions of the gene that are found in nearly all prokaryotic organisms, the same sequence. And then the valleys that are labeled with V's, these are called hypervariable regions. And they vary so much between organisms that they act like a barcode that can kind of identify the organism that they came from. And so if you target a certain region of the 16S gene that includes a hypervariable region, then you can get an idea of, or like a census of the prokaryotic organisms that are in your sample. This is just a high-level workflow for a 16S sequencing experiment. We call them amplicons when we're targeting a specific region. And especially just want to highlight the middle step here because 16S sequencing requires that you do several cycles of a polymerase chain reaction, or PCR, which just makes a bunch of copies of the specific part of the gene that you're trying to sequence. And the importance of this will become clear a little bit later. I'll come back and talk about the PCR cycles and this amplification of the gene. And first, this is a hypothetical example of the kind of data that you might get when you do a 16S sequencing experiment. And you see that the beginnings and the ends of all of these sequences in gray have the exact same primer sequences. So this is what you use to target the gene in all the organisms, because it's essentially the same. But then they all have regions that vary in between, and they're color-coded by which ones match each other. And so this region gives you information about what organism that sequence came from. And then the abundance of each of the color-coded sequences here gives you an idea of the abundance of the organism in the sample. It's somewhat correlated. The goal of a 16S experiment is to give a community profile, sort of like this, that identifies what organisms are present, as well as their relative abundances or prevalence in the sample. And of course, we want this census or profile to be as accurate as possible. So the simplest way to go about getting a profile might be to just identify all the unique sequences in your sample and then count up the number of each. And that would give you an idea of the number of organisms and their abundance. And then you could map them to a database of known 16S gene sequences to get taxonomic labels. And while this might have worked well in the early days of sequencing, for example, Sanger sequencing, which was relatively low throughput, with modern high throughput sequencing technologies, this turns out to be a pretty lousy strategy because the 16S read sequences that come out of the sequencer are going to have a lot of noise in them. And so what this ends up meaning is that if you try to do this strategy with a modern sequencing experiment, you might think your sample has thousands or even tens of thousands of distinct organisms, and it might actually only have a couple dozen. Okay, so what do I mean by noise? When I use noise here, I just mean a sequence that's found in your read data set that is not found, that doesn't come from the biological sample. It doesn't match what's in the biological sample. So there's two different types of noise that are found in the data. The first is biological noise, And this refers to DNA contaminants. So it's biological DNA, but it's not in the community that you're studying. And this can happen as a result of contamination during sample collection or laboratory processing. There are a number of sources in the lab that can possibly contaminate the sample, and it's very difficult in practice to have perfectly aseptic technique. The other type of noise is technical noise. One source is from PCR, that polymerase chain reaction. It can happen where two biological sequences will spontaneously fuse in the middle to make a hybrid sequence that doesn't represent anything biological. That's called a chimera. The other kind of technical noise that I'm more focused on here, particularly with the Illumina sequencing platform. That's the predominant sequencing technology today. And the most common type of sequencer error in Illumina is when one nucleotide is misread and reported as a different one. This is called a substitution error. And so for my thesis project, I'm looking at different ways of correctly inferring the community composition when there's noisy data because of these different kinds of sources. Okay, so how do researchers deal with this? Well, for about a decade or so, the most popular strategy has been to cluster the read sequences into operational taxonomic units, or OTUs for short. And this uses a simple similarity threshold to cluster the reads. And so I'm going to illustrate how this works with a little demonstration. So I have a toy microbiome sample with these 5 different colored organisms. And the distance between the dots represents the difference between their 16S gene sequences, and the size of the dot is correlated to the abundance or the prevalence of that organism. So if I took this sample and did a 16S sequencing experiment, I'm going to get some noisy data that might look like this. So most of the read sequences are going to be the correct ones that are the darker shaded dots. But then some of them are going to have errors, and that's shown by the lighter shaded dots of the same color. So they're derived from the darker shade, but they have some sequencing errors. And they're kind of distributed around the biological sequences or the template that generated them. And so the way that OTU clustering accounts for this is just by using a fixed similarity threshold that makes like a radius like this, and the letter T is showing you the length of the radius. But I think from this example, which of course I designed specially to show you this, you can see that there's some problems with OTU clustering. First of all, it can't resolve fine variation. So if two organisms have very similar 16S sequences, such as the blue and the purple, then OTUs may cluster them as one and infer only a single organism. But then the other problem is that they can, especially with high throughput sequencing, there can be sequencer noise that falls outside the threshold. So there's these light green dots that didn't get clustered with the dark green organism that generated them. And so they can generate false positives like this one that doesn't correspond to anything real. So of course, there must be an alternative. And one that's emerged in the last couple of years is to replace this fixed distance threshold with an explicit model of the sequencer error. And these error modeling methods, as I'm going to call them, take into account not only the distance between sequences or the difference between them, but also the abundance of their reads in the data. And so to illustrate with multiple dimensions, both the distance between and the abundance, I'm going to use a different representation. So instead of dots, I'm going to represent a sequence by a bar here. The Hamming distance on the x-axis, Hamming distance is just a measure of how similar or how far apart sequences are. It's how many positions are different between them. And then the height of the bar is showing you its abundance. And so this dark orange bar, let's say, is a single organism right now in my sample. And if I did an experiment, first of all, the dotted gray line is a hypothetical error model. And so what the model shows is that I expect the correct sequence to have the highest frequency. So that's where the peak of the error model is. It's centered over the true sequence. But then the more errors I have, so the farther away on the x-axis, the less frequent I expect those errors to be. A single error may be relatively frequent, but multiple errors is going to be rare. Okay, so what if I use this to cluster that same toy sample? Well, First, I need to transform it from dots to bars. And then if I did a sequencing experiment, I'll get some noisy data. And you see the errors in the gray bars there. And then if I apply the error model to this data set, this is what I might get. And you can see that the error model, the hypothetical error model resolves all 5 of the organisms in the sample because each of the true organisms is too abundant to fit under or be consistent with the error model of any of the others. And so it does very well, in this, at least in this example. And then for contrast, OTU methods will just bin these sequences like this. It doesn't consider the height, it's only a fixed width of the distance between them. All right, and there's one more sequence inference algorithm that I considered for this project. And this approach looks at the total amount of variation, which is also called entropy, at individual sequence positions in a set of reads. And so by a sequence position, I mean a column in the aligned set of reads. So starting with the full data set on the left, all in one cluster, if the entropy of a certain position, or the variation, is above a certain threshold, then the algorithm would split that cluster to make the entropy 0 in the new cluster. So you see all the sequences that had A's went into cluster 1, and all the sequences that had G's there went into cluster 2. And then this continues, so this column maybe has some variation, but there's only one difference there. It doesn't get above the entropy threshold, so we leave it alone. But then there's another one with higher entropy, several differences, and so it gets split. And once the entropy of all positions in the clusters is below the threshold, then your clustering is complete. So this is the entropy-based algorithm. So now that you have an overview of the algorithms I'll be comparing, I'm going to move on to talk more about the specific methods that I'm benchmarking here. My first aim, which is to look at how well they perform on high microbial biomass samples. So this kind of gives a baseline of their performance before I move on to the second aim, which is looking at how things change when we have decreasing microbial biomass of the sample. Okay, so these are the 6 different community inference methods that I'm comparing. There are 2 OTU-based methods, 3 error modeling methods, and a single entropy-based method. And I'm going to briefly discuss my choice of each one of these. So first, for OTU methods, there are more than a dozen published OTU clustering methods because they've been so popular for so long, and some of them have multiple versions. So first of all, I limited my choice to only methods that don't rely on a reference database for making their cluster seeds. First of all, the references are always changing and being updated, so it's kind of a moving target. But also, they introduce bias from the beginning based on which reference you choose. Also, the other methods don't use reference databases, so this allows more of an apples-to-apples comparison here. I chose the first one, uclust, because it's been the default method in a very popular 16S analysis pipeline called CHIME. And so CHIME has been cited in a lot of papers, so uclust has been sort of a de facto standard in the field for a while, even though I think in some cases people don't realize it because it's just the default method in CHIME. I didn't expect uclust to perform very well, but because it's been used a lot and cited, it provides kind of a baseline for comparison and how much better we might be able to do. uparse, the other one, is a very similar algorithm to uclust, but it's different in that it performs an additional quality filtering step before it makes the read. So it gets rid of a lot of the lower quality data before it starts making the clusters. And uparse, in a previous benchmarking study, was shown to reduce the number of false positives relative to most other OTU methods. Okay, now for the 3 error modeling methods. I chose all 3 of these because they're the only 3 standalone error model methods that have been published so far, and I think they all have the potential to improve on the OTU approach, as my demonstration probably suggested. To give you a brief idea of the differences between them, there's 2 of them that use an a priori model. So the model is fixed before it sees the data set. It's not based on a specific data set. It's just a sort of average estimate of the error. And then there's one data too that does look at the data set and sort of adjusts its error model based on the data that you give it. And then among the two methods that use an a priori model, UNoise uses a smooth parametric function that looks a lot like the hypothetical error model I showed you earlier. And de Boer uses a piecewise error model that specifies the expected frequency of errors at each Hamming distance as you get further away from the template. And finally, I chose MED because it's the only published example of an entropy-based method. And it's also been shown to have better sensitivity than OTU methods. And so it kind of gives an alternative to OTU and error modeling methods, so at least we have three. And then MED, besides what I already described about entropy-based methods, has this, after cluster formation, it attempts to filter out noise by removing clusters that have low abundance relative to the data set. And by default, any cluster with less than 0.02% of the reads of the full data set will get thrown away because it's considered to be just noise of some sort. All right, and then to benchmark these 6 methods, I needed some data sets where I sort of knew ahead of time what the community composition was. And so for this, I used what are called mock communities. And these are biological samples that are prepared by mixing isolated cultures of organisms in sort of fixed proportions. So I know what I'm putting in. I know what to expect in the community composition. And for the first aim, I chose, for the high microbial biomass, I chose 3 publicly available mock community data sets and 1 new data set that was generated here at OHSU by Mark Asquith's lab. So I've nicknamed the public data sets by their first authors. And the Kozic data set has 21 different bacterial strains, and they all have equal starting proportions of DNA. The Schirmer and D'Amore data sets have the same 57 strains in them, but the Schirmer has equal proportions of DNA, whereas D'Amore uses varying proportions or uneven proportions. And then the new data set called Zymo is from the Zymo-Biomics Microbial standard. And it has 8 strains with equal proportions of DNA. So this is a schematic of my data processing pipeline. The first phase in blue is where I filter out the very low quality data and trim the ends of the reads that are typically unreliable. And then the second phase is the meat where I do the community inference with one of the 6 methods. And the third stage is where I analyze the results and try to make distinctions between them. So the first and the third stage, with some minor exceptions, stayed the same for each of the 6 methods. And of course, I need to have a way of evaluating results. So, starting with the complete set of inferred sequences from each method, I classified the sequences according to a scheme that I'll show you here. First of all, if an inferred sequence perfectly matched the known sequence of one of the community members, then I called it a reference. And then, so the more references I detect relative to what I'm expecting, the better a method's sensitivity. So this is a measure of sensitivity. If it's 1 to 10 nucleotides different from a known reference sequence, I called it ref noisy. So the idea here is that these are most likely sequences that are the result of sequencer error, technical error, in sequencing the reference sequences that were misinferred as distinct organisms. So any sequences that were remaining, I compared to the NT database from NCBI. And this is sort of like a giant catalog of known DNA sequences. And any remaining sequences that perfectly match an NT sequence, I called a contaminant. The idea is that these are most likely true biological contaminants if they match a known DNA sequence that I wasn't expecting. Anything that is 1 to 10 nucleotides off from a contaminant sequence, I called contam noisy. Again, the assumption is this is coming from a sequencer error in trying to sequence these biological contaminants. And finally, anything that's left over is just other. It's none of the above. And I also want to point out that ref noisies in particular and contam noisies to an extent are measuring the specificity of an inference method, because I want, ideally they'll have very few of these because these are from technical noise, and I want them to distinguish that technical noise is not a real organism. So if there's a bunch of these, that's not good. That means low specificity. All right, now without further ado, I'll show you how each of these methods performed on the high microbial biomass mutt communities. Okay, so first of all, I think it's pretty interesting just to look at the total number of sequences inferred. So each row in these tables is corresponding to one of the 6 methods, and each column is one of the 4 data sets. And the rows are color-coded by the type of algorithm also. So one thing that stands out for me is that UCLUST is always inferring the highest number of sequences among all methods, and sometimes by an order of magnitude. UParse is tending to be on the high side as well. The 3 error model methods down here are generally reporting lower numbers of sequences, and they're kind of in the same ballpark as the expected number. The expected community sequences are on the bottom here in bold. So the error model methods look like they're closer to what I expect, at least. DBlur is always reporting the least number of sequences, and Data 2 is on the high side for the error model methods. So to get more insight into these total numbers, we can look at the classification scheme that I applied. So starting with the reference sequences, we find that even though the OTU methods reported high numbers of overall, they have relatively poor sensitivity because they're always detecting the fewest number of community sequences or references. And the error model methods are typically detecting the most reference sequences. The OTU methods are especially bad on these more diverse data sets where they have quite a bit fewer reference detections than the others. Among the error model methods, it looks like DBlur is maybe a little less sensitive than UNoise and Data 2, especially right here, you see that. And no method is perfect on the DAMORE data. So it's possible that some sequences were so rare in that data set that they may have been removed by the quality filtering step or just not shown up in the data set at all. All right, next, the ref noisy category. It also shows some big differences between the methods. Here we find that only UCLUST and MED have notably large numbers of sequences. And again, these are supposed false positives. So this captures sequences that have errors that were incorrectly inferred as distinct organisms. And this suggests that UCLUST and MED are not dealing with the sequencer noise very well. But UParse and all the error model methods don't seem to be affected by this sequencer noise much at all. Okay, and the contaminant category was supposed to represent biological noise or contaminating DNA. And you see that pretty much all methods report at least some contaminants. So I'm concluding that there is some biological noise present in each of these samples. You can see again that the OTU methods are reporting relatively high numbers that I've highlighted here, and MED is reporting the fewest. So given that we've already seen that the OTU methods were less sensitive and in the case of UCLUST less specific, I don't, with the reference sequences, I don't take this as meaning that these are more sensitive now. I think this actually shows poor specificity on the part of the OTU methods. They're finding false positives in the contaminant category here. And MED's very low numbers here are mostly explained by its noise abundance filter. So the contaminants are at a low enough abundance that they didn't pass the threshold for MED's noise filter, and they just got thrown away. Among the error model methods, you see that Data 2 is reporting the most. So that either means that Data 2 is more sensitive than the other two, or it could be less specific, and these might also be some false positives. But I don't think there's enough evidence to decide either way right now. And then finally, there's the other category that shows some difference. It mostly just shows that UCLUST and UParse apparently these are probably false positives. I can't be totally sure, but they're the only ones that have notable numbers here. And so it's further evidence that they are overestimating the sample diversity, I think. Okay, so far I've just been looking at the number of unique sequences that are inferred by each method. But if we look at abundance, or the number of reads that are supporting the sequences in each category, we get a pretty different picture. So here I'm showing the total relative abundances of sequences in each of the 5 classification categories. And so there's a different plot for each of the 4 data sets, and then each of these bars is one of the 6 methods, as you see on the x-axis. So here we see that the vast majority of reads are supporting the blue reference category. The plots look pretty boring because it's all reference. And that's the target community. So the only non-reference contributions of note are these light blue ref noises, the biggest one for MED and a little less for unoise and data 2. And then we also see that for the Schirmer and Damore data sets, it looks like all methods are inferring a small but notable proportion of contaminant sequences. That's the red. All right, so I think we can make some broad conclusions from this first aim. The OTU methods and MED all showed either poor sensitivity or poor specificity or both. And all 3 of the error modeling methods had pretty good sensitivity and specificity on all the data sets. So looks like the error modeling methods are kind of the clear winners here. Also, there were a few contaminant sequences detected in each of the data sets, but their overall abundances was very small, and so they have a relatively low impact on community inference. So that brings me to my second aim, which was to evaluate sequence inference when the microbial biomass of a sample is decreasing and getting small. Okay, so how would low biomass change the overall picture? That's the question. So as we just saw with high biomass samples, there is some noise from bacterial contaminants. But as I'm illustrating, the level of contamination is pretty small relative to the actual community sequences. And the sample sequences in blue dominate the read data set. But one thing you have to realize is that even if the community biomass gets smaller, the contaminating DNA is not going to get smaller in the same way because it's coming from an outside source. It doesn't come from the community sample. So when I go to amplify the 16S genes in PCR, like I mentioned earlier, if there's less community DNA, then the contaminant is going to make up, the contaminant DNA is going to make up a larger proportion of the total sample, and now it's more likely to get amplified in PCR along with the community gene sequences. And so if you get a very low biomass community, you might end up with a picture more like this, where the contaminants get amplified and sequenced enough that they actually make up a large proportion of the overall sample in the data set that you're sequencing. So I'm kind of looking for 2 different things in the second aim. How is community inference impacted by this scenario? How much of an impact does contamination have? And also, how do the different methods handle this situation, and what does their performance look like when we have low biomass? So to mimic samples of different starting biomass, we created a dilution series. Again, this was in Mark Asquith's lab here on campus, from that same Zymo mock community that was used in AIM 1. So a total of 9 samples were generated by diluting each successive sample to 1/3 the relative dilution relative to the previous sample. And so that gives you a dilution series with relative dilutions that are shown in this table here. So the DNA concentration is a stand-in for microbial biomass of the community. And then these 9 samples were sequenced on an Illumina sequencer to get the raw data. And this undiluted sample here, it's the same one that I used in the high biomass first part of the project. And the procedure for the second aim is essentially the same as for the first, except now instead of benchmarking methods on different data sets, I'm using the same mock community, but now different dilutions. And of course, I'm using the same classification scheme to evaluate the results. So with that, let's see what I found on the dilution series benchmarking. And I'll mostly go through the results one category at a time, at least the interesting ones. But instead of showing you tables, I think it's easier to see visually like this. So now along the x-axis, you have the relative dilution. So this is undiluted, and this is the most dilute or least concentrated sample. And the y-axis is showing the number of sequences inferred by a method. Methods are color-coded in here, and also they're color-coded by algorithm. So OTUs are blue, orange for MED, and error models in green. So starting again, these are the total numbers of sequences inferred. And the trend that I think we see pretty obviously is that for a given method, the number of sequences inferred tends to increase as dilution increases. And hopefully that makes sense given my discussion of what happens with PCR amplification. And at lower dilutions, we see, just like with high biomass, UCLUS is inferring the most sequences. But then when we get to higher dilutions over here, it's no longer the OTU methods, but it's some of the error modeling methods, and MED gets to the highest of all there. And it looks like the sequence totals drop off a lot at the end of the dilution series. The main explanation for this, I think, is that those samples had a much smaller sequencing depth, so they just generated a lot fewer reads. So they're around 50,000 reads compared to 100 to 200,000 reads or more for most of the other samples. So if this were corrected for the number of reads, then this trend would pretty much continue across the dilution series. All right. All right, this is not a test of the emergency broadcast system. This is actually showing the number of reference sequences. And what happened here is that all the methods recovered all of the community sequences at each dilution. So it's kind of disappointing because I thought that at least some of the methods would have imperfect recall over at the higher dilutions. But my conclusion is not that they all had perfect sensitivity. It's just that this particular mock community didn't challenge the sensitivity of any of the methods well enough. But if we move on to the ref noisy sequences, there are some interesting differences. Just like with what we saw with the high biomass samples, we see that UCLUS and MED are not doing a very good job of recognizing sequencer error as noise. But the other four methods that have very low totals of ref noises across the dilution series, they don't seem to be affected pretty much like we saw before on the high biomass. But none of this explains why the sequences are increasing across the dilution series until we look at the contaminant category. And so here we see what's really driving the increase in total sequences inferred, because there's a pretty dramatic increase, especially from the 1/3 to the 1/9 dilution and then continuing on. And you'll see that the y-axis is going above 300, so it's a lot of purported contaminants when the concentration is dropping. So to make it easier to see what's going on with each method, I'm plotting trend lines for each of them separately. So there's six plots for the 6 methods. Contaminant counts are in orange, and contaminnoises I also included in purple here. So some important differences show up. First of all, for the 3 error model methods that are on the bottom, you see an almost linear increase in the number of sequences inferred as the concentration decreases. And going back to my discussion of what happens when biomass gets low and the contamination is increasingly amplified, then I think this linear increase in sequences detected is what we should expect. And so I think it's evidence that the error model methods are doing a reasonably accurate job of detecting that biological noise. By contrast, the OTU methods, these first 2, they show an initial jump in the number of sequences detected, but then it kind of levels off and plateaus, and they stop responding to changes in the sample dilution. And so this, to me, I think this shows a lack of accuracy. They're not super sensitive right here. I think this is more lack of specificity and false positives, and then just not responding to dilution is more evidence of poor accuracy by the OTU methods. And then MED is kind of all by itself. At first, it doesn't report any contaminants, and then it jumps up to show the highest number of all. At the beginning, the abundance filter is taking out the rare contaminants. And then when they get more and more abundant, they get over the filter. But because it spikes up so high, I think this is more evidence that MED has poor specificity. I think there's some false positives that are inflating this number, which is also what we saw with the ref noisy sequences. So again, those are the main results for the sequence counts, but I also want to show you what's going on at the read level. So there's not a whole lot of difference between the 6 methods here, so you can kind of just pay attention to one of them. The most striking thing is that as the concentration decreases, the contaminant reads are making up an ever-increasing proportion of the total sample. So not only do we see that the number of contaminant sequences increase, but also their overall abundance in the sample is increasing, and they're actually really dominating the sample at the lower concentrations. All right, and finally we can get a different kind of information if we look at the distributions of the abundances of the sequences at each dilution. So I'm showing here log transformed abundance distribution. So note the y-axis, it's in powers of 10. So a small jump actually represents a big difference. And there's just 2 groups that I'm plotting here. The reference abundances in blue, abundance distributions, and the contaminant abundance distributions in orange. And I only plotted 2 methods because I don't have room on one slide for all 6, but also you have to believe me that the trend we see for deblur is pretty similar to what I saw for most of the methods. So it's representative. And the plot gives you a sense of how well the sequence inference can separate the target community from sources of biological noise. What I found for deblur in most of the methods is that as concentration decreases from left to right, the reference signal distribution in blue is kind of steadily declining. And on the other hand, the abundance of the contaminant sequences on average is rising as we go to lower concentrations. And so they go from being pretty well separated to hopelessly overlapped over here. And so this shows that for low microbial biomass samples, we can't hope to separate target community from biological noise by using just a threshold filter. Anywhere we draw a horizontal line here is either going to cut out some of the reference sequences or include a bunch of the contaminating sequences, which is what we want to avoid. And then MED is the exception to this trend. You can see that the contaminants are relatively flat. Again, this is probably mostly due to its abundance filter, which is cutting out all of the low abundance sequences at the bottom and flattening out that trend. But to me, the most interesting thing maybe from this trend is that it kind of suggests a possible strategy for identifying and removing contamination. And the idea goes like this. If you include a dilution series as a positive control with your study samples in a 16S sequencing experiment, then you could look for sequences in the dilution series that have a positive correlation with dilution, and you could label these sequences as contaminants. So you see the orange highlighted ones have rising abundances in the dilution series. And then you can remove any of those sequences. You get a set of contaminants, and then any of those sequences that show up in your study samples that are labeled as contaminants, you can just get rid of them. So this is not a totally original idea. There is a version of this that's been implemented by another group, but there's certainly a lot of room for research here and new ideas or implementations. All right, so I think we can make some general conclusions from the overall study. First of all, a recap of the high biomass. We found that UCLUS, UPARS, and MED all suffered from either poor sensitivity, poor specificity, or both. So probably less recommended to use these, whereas the error model methods had reasonably good sensitivity and specificity. Also, we saw that contamination was present, but at a pretty small abundance, and so maybe not a big impact on community inference. But with the second aim for the decreasing biomass, contamination looked like it can have a very large impact on community inference overall. There are many more contaminant sequences that show up, and they tend to have much higher abundances and make up a large portion of the sample. We also saw that no method on its own can simply tell the difference between reference sequences or community sequences and contaminants. So, so I'm more interested in which ones are accurately identifying the contaminating sequences, which looks like it was the error model methods, because they showed the best correlation with concentration, both in terms of the numbers of sequences that they inferred, but also the abundances, which all methods did pretty good with. But one thing to note is that I couldn't really make much distinction between the 3 error modeling methods, so I think further benchmarking research with more data is needed to kind of determine which of those might be the best in this situation. And finally, more research overall is needed in the area of contamination identification, especially for low biomass 16S sequencing experiments, because it really has a large impact, and for the most part, I don't think it's not being addressed adequately by the current research. And I just want to finally point out a couple of limitations for this study, some caveats. First of all, the mock community structure. You saw with that ugly plot of the reference sequences that the composition of this mock community, the Zyma mock community, is not necessarily representative of a biological sample. It's only 8 sequences, and typically you would have more different organisms, and they also wouldn't have the same relative proportions to each other. There'd be a wide range of abundances. Also, the classification scheme that I used, the definitions are inexact, so inevitably there will be some overlap between the different categories. For example, ref noisies and contaminants might get mixed up a little bit, but I think overall the results that I saw pretty well supported the definitions that I used and made useful distinctions between the methods. And finally, I only had 1 dilution series, so especially for distinguishing between the error modeling methods, having different dilution series from different mock communities would be nice to help make more quantitative distinctions in that area. All right, and finally, I want to thank all the people who have supported me in developing and executing and persisting to the end of my thesis project, especially Lisa Karstens, my advisor, the rest of my advisory committee, Mark Asquith, Guanming Wu, and Shubo Song. Thank you all very much for being on my team. Also my family, some of whom are here, my girlfriend for putting up with my antisocial behavior for the last year or more, and Shannon McQueenie for helping me with the early development of my ideas, and all of you for being here. So thanks, and if you have questions or critiques or anything you want to comment, I think we have some time left to take it, hopefully.
Thank you. Thanks a lot, Vincent. Very nice talk. Just one critique I had is one I've mentioned before, is that your benchmark, if you like, is number of sequences, isn't it? So presumably 2 sequences could come from the same bug, right? The same species of bacteria. So from a biological standpoint, do your 100, say, contaminant sequences come from 10 bacteria? Do they come from 100 different bacteria? Which is pretty important, right, when we think about the ecosystem that's there.
Yeah, and I guess I think that it depends on what the goal of your experiment is, because sometimes only broad distinctions are important. I mean, that's certainly the extension into the biological realm, and it's a valid point. I think sometimes the differences are only important at a broad level, and then you know, a lot of the sequences that I'm showing these methods detecting might be grouped into many fewer. But also, I've read benchmarking studies that are suggesting that organisms that are of the same species but have a very small difference in their 16S sequences, so that they are purportedly from different strains, actually occupy distinct ecological niches. And so if that's something that's important to your research, then I think that even the very fine sequence differences might matter.
Okay, and one more question while I have the mic. So it seems that DeepBlur and Data2 are probably the best algorithms that you tested, more or less, you know. And I was wondering if you could comment on the ease of implementation of those two different algorithms. Like, which do you think would be, is there one that's clearly much easier than another, since you've implemented both?
I think the most obvious difference between them in terms of use is that dblur is quite a bit faster to run than Data2, because if you remember, Data2 uses, it estimates its error model from the data, and that estimation step takes a long time in the algorithm. And dblur uses a model that's specified beforehand. So if you have limited computing resources, then dblur might be your friend. Also, dblur has more of a, I don't want to be derogatory, but it's a canned pipeline where you can call one command and it sort of runs the whole thing, whereas Data2 is implemented in R, and you have to use several commands in an R script to properly execute the Data2 pipeline. So I'd say dblur is a little bit easier and faster. Eric over here.
Awesome talk. I really enjoyed it. Curious about the contaminants that you found. Did you investigate what those were by any chance?
I did a little bit.
Yeah, that might be interesting.
Yeah, I would have liked to do more digging into distinguishing what was there.
Yeah.
Mostly what I saw is that the vast majority of them are bacterial and prokaryotic, but somewhat surprisingly there were a handful that hit the NT reference that were not prokaryotic. I remember a bunch of them matching various species of camellia.
Okay.
But they were at very low abundance, and I looked at those sequences and they barely showed up. But I think it's even possible that, you know, in a lab that, you know, if it's close to a flower bed outside, something could sneak in there. Who knows? But no, I'd like to look more into that, and that would be a way to take this project further and look at what those contaminants really are and how confident we might be that they are biological.
This kind of dovetails on that question, but I get asked all the time in a different context about something about cancer, like so contamination in terms of inference of somatic mutations. I'm just thinking in terms of guidelines. Like you did a very nice job of the algorithms in terms of sensitivity and specificity in AIM-1. But one of the questions is, for a lot of people, they're not going to know how contaminated their data sets are. So do you feel comfortable kind of with thought for the community in the microbiome community about that, or are you uncomfortable? I guess it's just a question, you know. And I'll give you a preface that in the cancer community, we know that the level of contamination will vary patient to patient. So I'm not necessarily. I'm just curious your thoughts after doing all this about that.
So if it varies patient to patient, maybe it has something to do with sample collection?
It absolutely does. I'm just thinking for I'm making that analogy. So I'm just curious for your thoughts with the microbiome work, you know.
So in a real sample, it would be, you couldn't apply the classification scheme that I used to a sample that wasn't a mock community, because you wouldn't have any idea what the references were.
I was thinking more about the methods. Like, some of these questions where people are saying, you know, you're showing that the inference methods are sensitive to the level of contamination. And so I was just thinking as a guideline to the community, given that I'm out there doing work in this area, and I'm trying to leverage the things that you're doing. I'm just wondering if you feel comfortable, which you don't have to, making a statement about that in terms of what you might say. Because we're often asked this. This is the question. Yeah. What method should I use? And so I'm just curious your thoughts after doing all this work.
Well, I think I would still feel much more confident using an error model method for inference of anything, really. I think OTU methods are, you know, probably on their way out, and more sophisticated algorithms have been developed, and the error model methods and their results show that. But I don't think that using those methods on their own without some further development of an algorithm to identify the contaminants, which is what I was sort of suggesting at the end there, those methods on their own are not going to tell you how contaminated your sample is. We need more research into identifying something like a trend over a dilution series, for example. From the data that I've looked at, it looks like that trend shows up a lot in those contaminating sequences. So I think that there's a way of, I think there's a formula you can come up with that would give you a pretty good sense, but that's further research that I'm pretty interested in looking at. Maybe we will.
So for the error model methods, one approach is to use a prior model that's already predetermined. Mm-hmm. The other one is where the model is estimated from the data. I wonder, using this mock community and knowing at least the information about the references, if not the whole ground truth, could that knowledge help you in return to have a better estimate of the error model for the approach that's actually using the non-prior approach?
Like the data 2 that estimates from the data? So if I understand your question right, I think another extension of this that I would like to do is looking at mock communities that have different error profiles. If you look at the output of the sequencer error, it tells you, the sequencer will tell you its confidence in the quality of its data. And so if I use that quality information and I have data sets with different qualities, I'd like to do a study that looks at the method's performance across different quality profiles and see how, if there's wild variation with quality or if it stays pretty consistent. My guess is that data 2 would do better in that scenario because it does look at the data and use that quality information in estimating its model. And I think that was even somewhat apparent. I didn't want to point it out because it was kind of a side point, but there was, let's see if I can go back far enough. Yeah. So here, I think there tends to be much more variation between methods on the Zymo and Kosich data sets in the number of sequences inferred. And one thing I didn't mention is that these data sets do have lower quality profiles than these two over here, and I think that helps explain this wider variation. But yeah, and you do see it in the models, although it doesn't look like data 2 is doing better. I think there's another reason for that, but it's kind of getting into the weeds.
Sorry, Vincent, for all the questions. One more. I was just wondering, do you know for those 4 different mock communities, were similar DNA extraction protocols used for them? And then sort of second part to that question, did you see any overlap in terms of sequences that might be common lab contaminants that you might, say, find in 4 different labs, maybe because they're using the same extraction kit or whatever?
I don't know off the top of my head what the extraction kits were for the 4, so I can't comment on whether they were the same or not. I can say that I was shocked by the level of quality in the sequencing data from Schirmer and Damore. It looked like it was really, really, really good, and I've seen now a lot of Illumina sequencing runs, and I rarely see that profile. So it's possible that it was filtered before it was published, but I don't even think that's the answer. Sorry, what was the second part of your question? Oh, right. So I did classify sequences that showed up in all the data sets, but then I didn't summarize it for the talk, and so there certainly are some, but out of, there was over 300 contaminants detected at certain dilutions, and so certainly there's some of them that show up in all the data sets, but I'm not sure if that's meaningful particularly. But I'd be happy to look at it more for you later.
Just as a comment following up on that, that actually would be quite interesting, though, for your actual thesis or for the publication, because we actually can learn a lot from that if it is certain ones that keep coming up and all the methods do well, or some methods do different. So that additional piece could actually be really valuable to the community. Yeah,
and I guess my assumption also is that that's going to tend to be a batch effect, depending on the lab where they were processed.
Could be, but you won't know until you.
Yeah, and other papers have taken that approach of just trying to catalog common contaminants, but I don't think it's been done across enough study centers or labs. for that to be a confident database.
Okay, so we unfortunately need to cut off questions for time. So for friends and family who would like to wait for Vincent, we can ask you to wait out in the hall because we need to clear the room. The committee is going to have a closed session with him. So thank you very much. And if you join me one more time in thanking him for it.